problem when it comes to a eukaryote transcript is that eukaryote chromosomes are very long. They've been evolving, depending on the lineage. Like eukaryotes themselves appeared billions of years ago. And weirdly enough, eukaryote chromosomes they happen to collect what has in the past been referred to as junk DNA, and generally better referred to as non-coding DNA. And all this DNA gets collected and it gets scattered throughout the genes. So basically, if you look at an RNA transcript, right, you're going to have a bunch of coding regions and then interspersed amongst them are going to be non-coding regions. Now, we of course can't just use coding and non-coding. We need vocab. So our non-coding regions are going to be officially referred to as introns, and our coding regions are going to officially be referred to as exons. And we can't just make a protein using this transcript, because if we do that, if we feed this transcript into a ribosome and just try and make a protein, those non-coding regions, those introns, they're going to mess the whole thing up. It won't have the proper functionality, which means the first step in RNA processing is getting rid of introns, cutting out introns, and splicing together exons. What we need is an all exon strand of mRNA. And to do that, we're going to need the aid of an adorably named chemical. And this adorably named chemical is the snRNP. Now, remembering that, kind of annoying. But knowing its actual name, knowing the name that it is widely used in publications and papers is pretty adorable. snRNP is also known as a SNRP. So what do SNRPs do? SNRPs bind to introns. And what they end up doing is causing those introns to fold themselves and then eventually get cut out of the strand. That zoom out thing keeps happening because when I go to click a different color in this, apparently it decides to change the zoom level for these tools, and it immediately switches to either this one or sometimes the magnifying one. Very annoying, but this is how the SNRPs do. So SNRPs, they bind to introns, and then they fold the introns up, and they cut the introns out, and then they splice the exons together. And then what you have is an all exon transcript. And an all exon transcript is going to make a proper protein. Is this mRNA? Unfortunately, no. This is not yet mRNA. The next step in RNA processing is going to be protection. And that will be our next Biology Bite.